So last but not least, I invite now Professor Dina Fonseca of the University of Rutger to present some research work.
Hi, hello. I know I'm the only thing between you and lunch, so I'll try to make it really quick. First of all, merci, Jean-Philippe and Vincent for inviting me. I think this could be a fantastic operation. If we can make this work, there's a great group of people already, and I think we can make a big difference. Now, I'm going to sort of half my talk because in some ways, okay, how do I go forward? Yeah, I did that. All right, maybe we just, one slide. All right. Start over. There we go. Okay, so I'm going to change things a little bit here. We're going to be looking at insecticide resistance from the point of view of no matter what you do in your backyard, you may end up having insecticide resistance because it came from someplace else. So I'm a population geneticist. I look at populations of mosquitoes across the world. And so the one mosquito that we know is across the world is Aedes aegypti, which expanded worldwide probably quickly after, and the Portuguese, I'm originally from Portugal, sort of started exploring and then gave ideas to everybody else. So Aedes aegypti was moved with traffic of boats and slaves, the same way that the disease was, yellow fever, was moved around. So that was in the 1500s. Amazingly, it took until the 1960s for another mosquito to do that kind of sort of worldwide expansion. And that was Aedes albopictus. Now, Aedes aegypti is tropical. Aedes albopictus has both tropical and temperate populations. And that's really why it got the name of the biggest, most invasive species in the world. Take that with a grain of salt. But yes, it's a very invasive mosquito. But besides that, more recently, we had Japonica japonicus, which expanded across the world in the 1990s. And so these are three mosquitoes that I work on, actually mostly albopictus and japonicus. And japonicus, probably most of you haven't even heard of it. But it is a worldwide mosquito, and because it happened in the 1990s, I've been following the distribution and expansion of this mosquito basically on a yearly basis and looking at the population genetics that way. So I would like to consider and would like to propose this species as both a model, a good model to try to understand how extension of mosquitoes happen, and also as a potential mosquito that we may need to be concerned about in the future. Because japonicus is what albopictus was 30, 40 years ago, and both of them are what aegypti was before we started moving it around. All right, so what are the critical questions that we really all need to ask ourselves? We want to know how frequent are these introductions, we want to know where they're coming from, etc. We also know in many cases when you're doing control, and you fail to control, was it because there was insecticide resistance or because there was a separate introduction that just brought in the species quickly after? How much do populations change after introduction? And this is something that has very impactful for any kind of modeling, because if you assume the mosquito is going to continue behaving the same way it always did, and it changes, like with insecticide resistance, you can really have a big surprise. What about changes in vector competence? So one thing is insecticide resistance. From sort of broader standpoint, you can also have insecticide resistance affecting vector competence, or vector competence changing just because of demographics or selection. And of course, vector competence is just a part of vectorial capacity. Specifically for Aedes albopictus, because I'm a basic ecologist, evolutionary biologist, I want to ask questions about, evolution in terms of diapausing. How often does it happen? And basically, ultimately, how often could something like this happen again in the future for some of these other species? So, sort of the genetic basis of that. Now, sort of quickly, Aedes aegypti, and please don't take me. This is sort of a quick this is where it is kind of thing. Aegypti has really been all over the tropics for a long time, and there's been some really nice work done by Jeff Powell's group, first with microsatellites, which are these highly repetitive and really fast mutating loci that are used for high-resolution genomics, and more recently, an Aegypti SNP chip, which is basically has 27,000 different SNPs that allow you to do quickly an analysis of population of mosquitoes. So by using these methodologies, Julia Brown and Jeff Powell's group basically was able to identify and really have really nice data showing very high diversity in populations of Egypti in Africa, and indicating that it may have really been one single domestication event, that it may have happened because of changes of aridity, expansions of the Sahara Desert, or association with these old civilizations in North Africa that led to a domestic form of Egypti that then spread across the world in sort of after the 1500s. And interestingly, it seems to have expanded first into the Americas, and then from the Americas across the Pacific, arrived in Hawaii in 1892. And sort of think about Hawaii. I've done a lot of work in Hawaii, a very interesting test place. 3,000 miles from everywhere in the middle of the Pacific, a little set of islands. And we had Egypti arrive there in 1892 after Culex cuenca fasciatus, which arrived in 1826 or so. But then across across the Pacific and into the Asia, which sort of created a really interesting, one of the fascinating sort of epidemiological questions of yellow fever. Why is there no yellow fever in Asia? You know, is that associated with the background of the mosquito, etc.? Interestingly also, although this is a tropical species, there are, and just recently shown with population genetics, persistent populations of Aedes aegypti in Washington, D.C., which was actually pretty good because there were hearings about Zika just about that time right there. It was actually in Capitol Hill that the population was identified in Washington, D.C. So a temperate population, underground most likely, that is surviving. And again, that's a pattern we're seeing in these domestic mosquitoes. So they may be in places you don't expect because they started to be able to use a habitat that it didn't initially. Now, Aedes albopictus, as I mentioned, has two forms. It originally really has tropical and temperate population. And so in the 16th century, it was already found in Madagascar, potentially because Arab pirates were moving it around. 1896, yep, Hawaii. It's unclear how this mosquito suddenly got there, but it wasn't until the 1960s that it really started expanding. It expanded across Wallace's line, so this is a mosquito that only happened originally west of the Wallace's line. 1962 got to Indonesia, then 1972 to Papua New Guinea, 1979 Albania, expanding across Europe, 1983 to the Caribbean, and 85 Brazil and the United States, Harris County in Texas, 1991 Africa. So we now have albopictus in Africa, and there's sort of been continuous expansions in Africa. Torres Strait in 2002, and finally 2007 in, I don't remember, Nigeria. Somewhere in Africa. I don't remember which specific introduction this was, but basically a mosquito that has been expanding both in tropical and temperate populations. And basically now we have Aegypti tropical, albopictus tropical and temperate, and really climate-mediated interactions between the two species are actually driving the distribution of the two species. So a lot of ecology going on in there, and also some sort of interesting sterilization of male albopictus mating with female Aegypti, which does infertile matings. So now to the next one, Japonicus. So Japonicus is a completely temperate, very cold adapted mosquito. It actually occurs in parts of Siberia. And Japonicus was only in Japan until, as I mentioned, 1998, when it was found in Ocean County, in just around where I was at the time at Walter Reed Army Institute of Research, and also in New Jersey and in New York. It has expanded across the entire United States, in the eastern United States, in about 30 states. And then 2001 was also found in western United States. In 2002 in Europe, in Belgium, and is now expanding across multiple countries in Europe. And yeah, you guessed it, in 2003, it got to Hawaii. So now Hawaii has all three Aedes species. And that's not a temperate place, Hawaii. It does have some pretty tall mountains, which is precisely where Japonicus immediately expanded, but it's also occurring at low elevation in Hawaii, interacting very interestingly with Cuenca fasciatus, the Culex species. So Hawaiian Islands has the distinction of having very quickly been a point of introduction for all these Aedes mosquitoes. So Japonicus sort of has the northern distribution here, and interestingly, New Zealand is the only country that doesn't have any of the three species, indicating they've really done a really good job of keeping invasive species at bay. It's a very important strategy in New Zealand. So, invasive species of Aedes, why do we care? We already talked about quite a bit about this, so I'm just going to go fast. But the point is, we all know about Aegypti, but albopictus became a primary vector of the mutant 226 chikungunya, is considered a vector of dengue, and there's just an outbreak of dengue in the Hawaiian Islands, driven by Aedes albopictus with over 300 cases. Has been found, one of the few cases where a mosquito actually has been found infected with Zika was albopictus, both in Gabon and in Mexico, and it's regularly found infected with all sorts of different viruses. The question really is, how good of a vector is it? And what about Japonica? Really, it's sort of a past suburban kind of mosquito, but it's moving more and more into urban environments. In Hawaii, it's become very urbanized. So has in Europe and parts of Germany. We're finding a lot of complaints about Japonica. Albopictus, to give you an example in here, is a mosquito that will bite humans. So this is an analysis done just looking at the blood in the gut of wild-caught mosquitoes. We do find them we do find them feeding on humans a lot of the time. They do feed on cats and dogs and other mammals if they're around. We see that is true also with Aegypti. In the few areas where people have done analysis, of blood meal analysis on Aegypti and albopictus, they have very similar distributions of blood meals. You can have albopictus be 100% human feeder if you collect them near houses. And you can find Aegypti feeding on pigs and cats and dogs. It's just a question of availability. But the big thing in the United States in particular is that these are incredibly nuisance species. And as we've already mentioned many times, people will take mosquito control in their own hands. And in a place like the United States, it can be pretty devastating if they really take it seriously. The company is just propping up, ready to apply pyrethroids every 3 weeks, every 2 weeks, every other day if you really want that to happen. So it really is important to come up with actual mosquito control, because if we're non-controlled mosquitoes, we are never going to control the vectors. That's just my own opinion, by the way. So in terms of the kinds of molecular analysis that you can do for Aedes, there's sort of a history. We've done allozymes, mitochondrial DNA, genomic analysis, microsatellite loci have been quite successful. More recently, red tags, SNP chips, and now gene capture arrays. These all basically are trying to come up with DNA subsets that we can compare across individuals. And the reason for this is that, in fact, the DNA is highly, highly repetitive. Chen et al. Just recently published, and it's a beautiful paper, a publication of the genome of Aedes albopictus, and they sort of looked at the size of albopictus. Albopictus has the biggest genome size of any mosquito analyzed, with almost 2 gigabytes gigabases of DNA. If you compare that with Drosophila melanogaster, 175 megabases, basically 1 order of magnitude smaller, also true for Gambi. Then you start getting into quinks, which have about a quarter. And Aegypti starts to be kind of in the same sort of ballpark. It's just a tremendous amount of repetitive DNA. It also shows very wide intra-species, meaning among populations, variation in the amount of DNA, so-called C-value, lots and lots of variation. A lot of this variation has actually been shown to be associated with things that could be associated with insecticide resistance, like the P450s. Tremendous amount of variability. Which is really a good repertoire for being able to deal with whatever it is that we throw at them. I'm going to go through this sort of not really going into the details. The bottom line is you can do analysis where you remove the repetitive DNA. And so that's what this is about. And then you can basically sequence. And then you can do low copy enrichment by basically picking those low single copy DNA targets and just put them on a bait, like a little fish hook. You put it on a magnetic bead that allows you to basically take the DNA that you're trying to identify and sequence and just hybridize it with these fish hooks and then sequence them. And that's what this is basically about. But back to microsatellites. I mean, we've done a lot with microsatellites. And basically, these are repeats of these single patterns. They're very easy to score. For Japonicas, we've done a lot of analysis with microsatellites. And because I was able to really immediately catch these populations the year after they were caught, I was able to identify clearly different signatures, that populations coming in. So we had one introduction in Pennsylvania, and one in Connecticut, New York, New Jersey, very different signatures. And you can also look at comparisons over time, where you look at it in 99, 2000, by 2004, 2005, you see that the populations are now mixing, they're really variable. And so we're finding that as a sort of a general pattern. We've also been able to do analysis across the native population in Japan and Korea. And what we found there is actually that there's already mixing in the native range of these populations of mosquitoes. So we have sort of a pre-adaptation of the mosquitoes that then sort of expand across the world are already kind of being moved around in the native range. And so it really doesn't matter where the mosquitoes are coming from. They're all coming with basically a single signature, but when they get into so this is the native, when they get to the exotic range, this would be New York, Pennsylvania, Washington State, this is Hawaii, Belgium, Switzerland, and this is Austria, yes. You really see that they have very different signatures. So the exotic populations develop really unique signatures really quickly. In some cases, you have populations like in Belgium that over the years just remain completely the same as long as you don't allow them to move. This was a population that was identified immediately after it was found. It was in a used tire yard. The population was left to be there for 10 years. It never expanded outside of that particular yard because it was being monitored. Finally, in 2012, they said, the heck with it, and they went in and killed it. It's gone. But they really never changed. But populations left to expand, sort of develop new signatures, and you see multiple introductions. We also see mixing among the introductions, as I mentioned. And in fact, if you look at these kinds of mixings really, really carefully, and we've done a lot of these high resolution with lots and lots of different populations collected across a single area within a single year. In fact, this was all done within a single month. What you find is that the population is not being driven, this invasive species is not flying around. This invasive species is only being invasive because it's hitchhiking on us. So we're seeing signatures where populations are being completed. The genetic relationship between populations isn't because of distance, it's because there's a road that connects the two locations. So this kind of analysis where you basically the mosquito are hitching a ride. This has been shown, we just showed this, which I really like this title, The Hitchhiker's Guide to Becoming Invasive, and also being done by simply looking at cars and seeing that these mosquitoes are expanding in cars, and also looking at networks of roads and seeing where they are expanding. This was work done by Andrea. So in terms of, I'm actually going to end right here. So frequent are introductions. Really common. I mean, if you base this on this mosquito where we were able to really see it happened multiple times. Is a control failure or introduction? Actually, we should be able to tell this because each population really very quickly develops its own signature. How much do populations change after introduction? Well, they become mixed. So we're seeing a lot of decrease in genetic diversity locally, but then across networks of roads and transportation, we're mixing these populations with each other and creating sort of locally elevated levels of genetic diversity. How likely is insecticide resistance to occur de novo? May be able to tell using these kinds of methods, but not the mechanism that is driving that insecticide resistance. Changes in vector competence? We can't tell with the microsatellites, they're neutral loci. Changes in vector capacity? No way. So really what we need to do is link genotype with phenotype. And to do that, we really need to do exactly what Jean-Philippe was talking about, which is using things like P450s, or actually specific loci, and do these kind of genetic capture arrays with specific loci, specific genes in mind, so that we actually can find out what is the pattern and what is the genetic baseline of the pattern that we're seeing. Okay, we've gone through this. To do that, first of all, what we did was actually compare now Culex, Molestus, and Pipiens to identify loci that would be interesting. We found 4 different classes, things like olfaction, blood feeding, digestion, innate immunity, and gut protection, different genes that we have now created into 2 sets of baits. We have a high-resolution set of baits with about 91,000 probes for about 29,000 megabases, which we're using to really understand in Albopictus patterns of distribution and evolution, especially in terms of diapause. And then we have a lower resolution bait set of about 3 megabases focusing on 514 genes that all have really good reasons why we decided to look at that gene. And that is my last slide. Thank you.